Hello and welcome to Delve Deeper, the MAST's 2024 webinar series taking you on a group dive into marine science research without the need for a snorkel, scuba tank or wetsuit. I am your host, Phil Bell-Young, and today's talk is Shaving Private Ryan, the battle between regeneration and calcification of the coastal sea urchin and the multiple stresses impact of ocean acidification and warming. So I'm going to go straight into introducing our speaker. So our speaker today is a marine biologist at the Scottish Association for Marine Science and is primarily interested in the impacts of ocean acidification and warming on marine environments, particularly at a organismal and physiological level. Their current research is on the effects of ocean acidification and warming on sea urchins and their ability to calcify and regenerate their tissue in multiple stressor environment. Please give a warm virtual welcome to Angus Taylor.
Thanks very much, Phil. So yeah, I'm here today to talk to you about some of the work I've been doing as part of my PhD, which I've undertaken here at SAMS, which is a partner of UHI, and I work in collaboration with the University of Aberdeen. And this work is funded by the Super DTP. So quickly, I'm going to shoot through how I got here. I took a bit of a longer way around to start my PhD. So I started with an undergrad degree at Heriot Watt, and then I ended up getting a job at the University of Edinburgh working in a biomedical research facility, focusing on zebrafish research. And I stayed there for a few years, maintaining many fish, before getting an incredible opportunity to go and work for the UN over in Monaco, an environmental lab. So it was just a slight change of pace from Edinburgh. This was a radioecology lab, and it specialized in using radio tracers to investigate kind of all sorts of environmental issues, like microplastics, heavy metal pollution, and what I went there to study, ocean acidification. And I was there for almost about 4 years before I got the chance to continue my studies here at SAMS, looking at the effects of multiple stresses on sea urchins. So we all know about atmospheric CO2. It's increasing at an unprecedented rate, around 40% since in pre-industrial times. And I'm not going to go into too much about that. I'm sure we all kind of know about that. But basically, CO2 accumulates, traps thermal energy, and heats up the planet. And this is called the greenhouse effect. And unless this changes, it will keep on happening, and it results in all sorts of issues. The polar bear there has lost his ice float, so ice caps are melting. There's dramatic changes to weather systems, and this affects kind of terrestrial and marine environments. I'm more interested in what's going on under the ocean, so I'm here to talk to you about that. Apologies to Mr. Polar Bear there. But if the warming of the planet is known as the CO2 problem, I want to talk to you about the other CO2 problem. So this is still caused by atmospheric CO2. Basically, in the ocean, carbonate, so CO3, can be formed into bicarbonate in the presence of hydrogen, or with the presence of calcium ions, can form into calcium carbonate. And this is required by all these biomineralizing organisms in order to kind of lay down their hard structures. So crustaceans and corals and bivalves. However, atmospheric CO2 reacts with water and forms a very unstable carbonic acid. And when this is formed, it immediately dissociates into bicarbonate. And this releases additional hydrogen ions. And this influx of hydrogen ions causes the equilibrium between the carbonate reaction to shift towards producing more bicarbonate and less calcium carbonate. So this is bad. And this production of carbonic acid and the release of these hydrogen ions cause a pH decrease in the ocean. And that's why we call this ocean acidification here. So historically, stresses such as reduced pH levels have been studied pretty extensively, both in situ and in mesocosm experiments. However, the focus has often been on kind of the direct impacts of an isolated single stressor. So here we have a review back in 2005 of OA on calcifying organisms, of ocean acidification there. We have a paper here that my supervisor was involved in that led the way to my research on sea urchins again. We have cuttlefish behavior and CO2 conditions. We have ocean acidification affecting coral growth. We have another paper I was involved in on macroalgae and dissolution rates in CO2 levels, and even things like, weakened mussel shells. However, as I just mentioned, these stresses are kind of occurring simultaneously. So for my work, the influx of CO2 is causing both a dramatic increase in sea surface temperatures in the greenhouse effect, but it's also causing a notable decrease in pH levels and this partial pressure increase of CO2. And partial pressure is just a proxy for the amount of CO2 in the ocean, effectively. So as marine organisms are experiencing all these environmental pressures at once, the multi-stressor impacts of this are becoming more relevant, and it's a very relevant research topic now and should be considered. From the literature, we know that individually, temperature and pH have impact on several similar kind of responses across a wide range of species. So from calcification and growth to respiration, metabolism, behavior. However, while in some cases we see like an inhibition of a response, so stunted development or something like that, in other cases we can actually see an improvement. So growth rates tend to get faster with like minorly increased temperatures, for instance. So when considering multi-stresses, they could be additive, so combined sum. They could be synergistic, which is the greater than the sum of the parts, basically, or they could actually be antagonistic, where one stressor is mitigating the effects of another. So there's a big question that we don't necessarily fully understand yet. And we also don't really know about the mechanisms of coping with these extended periods of exposure, such as, years and years or multi-generational exposure. I'm going to quickly run through the background of calcification. This is the biomineralization process that enables organisms to lay down their hard structures, such as skeletons, shells, spines. And this up here is part of the previous equation that was being shifted away from due to ocean acidification. So where carbonate reacts with calcium ions in the water, and this forms calcium carbonate. And in the form of calcifying organisms, this is in the form of calcite. So what we have are these calcifying cells, and they're found on the surface of these calcifying organisms here. And what they do is they pull in calcium and carbonate, and they elicit a response, and they elicit the reaction that produces calcium carbonate here. And that's how they then lay down this calcium carbonate developing body part or the structure they're building, and they do this repeatedly and build up these incredible calcified structures. The only other thing that is worth mentioning now is a very important concept to calcification, and this is the saturation state. And this is a measure of the level of calcium carbonate in seawater, and it's related to its capacity to dissolve or precipitate. So above 1, calcium carbonate is readily precipitated, and it can be used to form calcified structures. But as it drops below 1, calcium carbonate tends to dissolve, and then forming and maintaining calcified structures is a lot more challenging. So as previously mentioned, hydrogen ions are shifting the equation, the equilibrium of this equation, sorry. And basically ongoing acidification is lowering the saturation state and then making it much more challenging for organisms to produce the required structures. So it's time to meet the troops in this fight. So this is a sea urchin. It's an echinoderm. And specifically, this is a coastal sea urchin known as Psammechinus miliaris. It's a small species, about 30 millimeters in test diameter, and it's also known as the green sea urchin. So this here is Private Ryan, and he is just one representative of many, many individuals that I've used in my experiments. So why sea urchins? Well, they're a widespread coastal species. They experience dramatic conditions, fluctuations daily with the tides, and they're living in these highly dynamic systems. They've been shown to have extensive regeneration and calcification, and this makes them an ideal model for me to be comparing these processes. So they have pentaradial symmetry, as you can see in the top left-hand corner there, with spines encapsulating the entire bodies. And they also have these 5 sets of 2 rows of tube feet. And these are fleshy appendages that form part of the water vascular system. And they have these adhesive suckers at the end. These tube feet, as you can see in that schematic, have a single nerve in red running up each one. And these are used for locomotion and sensory perception, and they can regenerate these entire structures, nerves included. So with the low metabolic rates and this magnesium calcite, which is a form of calcite with a deformed lattice structure, so some of the calcium is actually replaced with magnesium, and this makes it more soluble. But they could potentially be considered as a first responder for acidification and dissolution with the more soluble structures. I'll now quickly go into some of the work I've been doing here as part of my PhD. So this is a series of ongoing experiments that I haven't quite fully analyzed yet. So unfortunately, I'll only be presenting kind of some of the key things I've found and drawing some of the more obvious conclusions with some observational things thrown in for good measure. So we ran 3 main experiments here, the first being a temperature performance curve experiment, followed by an experiment looking into the initial wound healing and the molecular process of regeneration at early time points. And that was followed by a much larger multi-stress experiment to look into bringing these two things together and looking at regeneration and calcification across a variety of temperature and pHs. So first up is temperature performance curves here. So this first experiment was designed to assess the kind of regeneration of tube feet and the calcification of the spines across a wide range of temperature conditions. So we set up these water baths with individual housings for each sea urchin. And these are fed from header tanks that flow directly into the water, into the individual housing before going into the water bath and going to waste. So basically it was a flow-through system with no cross-contamination between individuals. So we selected our temperature range, and this was between 4 and 24 degrees. And this was based on the geographical kind of distribution of Psammechinus species. So we took the lowest recorded temperature in the coldest region they found, and vice versa for the warmest. And this showed a range of between 4 and 19 degrees in the wild. But as this was about ocean warming, we also threw in one warmer treatment for good measure at 24 degrees to ensure it was a stressor and hopefully to push them past the tolerance limits. So we performed a phenotypic assay, and if we think back to my main title, we all know what happened to Private Ryan. We shaved him, or more accurately, we amputated just one ambulacral section. I hope you can see in the picture there. And then we allowed them to regenerate until they were indistinguishable from the other non-regenerating sections. Non-regenerating from now on, I'll probably term as homeostatic. So in these studies, it took about 49 days, so 7 weeks to regenerate fully. So this assay involved weekly measurements of regenerating and homeostatic spines and tube feet using calipers and images and image analysis. And we measured homeostatic appendages as well to account for the naturally quite high variability between these individuals. So we let them regenerate. By day 21, they were well on the way to coming back, and by day 49, as you can see, hopefully, they were almost indistinguishable. They were almost fully regenerated back to normal. So what we expect results-wise from a temperature performance curve typically looks like this. It's got this nice gradual increase with temperature running along the x-axis there in performance. And this continues to increase until you hit the optimum condition, so T0 there at that plateau. And this is usually followed by a very dramatic crash of performance, just as you surpass the thermal tolerance limit. In my experiment, what we got was a bit of that curve, we think. So we didn't quite get to that point. There is a hint of a plateau coming from 20 to 24 degrees there. Statistically, there was no difference in those two warmer treatments. So we think we're about there, maybe slightly before the optimum performance. There was a very significant difference between the colder treatments and the warmer treatments in both spines and tube feet. And these regeneration rates measured here were calculated using a very typical growth equation. And that factored in the homeostatic appendage lengths too, so the maximum that they could achieve. And then we developed these K values up the side there, which are a proxy for regeneration rate. So the main question I had from this was, if they can grow so well at 24 degrees, why don't they live above 19 degrees if they can thrive there? And it seems like they don't. So growth may be expedited at these temperatures, but that's a very small part of the story, and there still could be a lot of stresses in other ways, sort of physiological and immune responses. So we also took writing responses, which is the time taken for a sea urchin to write itself when you put it upside down, a classic measure of fitness. And we also took celomocyte counts, so immune cells, to look at immune responses to stresses. These aren't fully analyzed yet, so that's sadly a case of watch this space for now. Following that, I took a bit of a jump into a second experiment, which is visualizing the early stages of tissue regeneration. And basically, this experiment was to look into the cellular processes surrounding the early stages of regeneration. So this was done much like the last one through an amputation of a section and a period of regeneration. But instead of looking at how it regrows, we then sampled these sections of tube feet, decalcified them, and conducted histology on them. So we stained and imaged them. And so this here is a homeostatic tube foot, so it's non-regenerating. You can compare it to that microscope picture there, hopefully, and see how it kind of translates from the structure itself into this histology image. And there's a few key characteristics here that I'd like to point out. So there's these I'm hoping you can see it reasonably clearly. These densely packed cells in the kind of corners at the top left and right of the sucker. And there's also this very well organized epithelial layer right at the top there. And these are quite key of homeostatic tube feet, as well as this well-defined kind of blue collagen layer and this muscle layer in dark red going through the sucker. The empty space in the middle there is exactly that. It's an empty space where the tube foot would typically be water-filled because it's part of that water-vascular system. And you can see these ridges along the edge where the tube foot would extend and contract with the muscles as required. So when we sample during the regeneration, we do start to see how kind of this regeneration process occurs. So day 0 was the wounding day. But by day 2, we had this classic epithelialization with the tightly packed cells, and this well-defined thick collagen layer had already been formed. By day 7, it looks like a blastema had already formed and differentiated. And a blastema is very typical of regeneration. It's a mass of kind of undifferentiated stem cells that would accumulate at the wound site and differentiate as required. However, I think I'm missing a key time point here that I have sampled and not imaged yet to look at this actual blastema formation. You can also hopefully see at CI there that cells are actually traveling between the inner and outer layers of this blastema area. Then by day 10, we had these classic tightly packed cells that I pointed out before that were characteristic of the corners of that homeostatic sucker. And this suggests kind of that growth develops there and then fills out in between to form the sucker. And from day 13 onwards, we have this fully formed and apparently completely functional miniature tube foot that just undergoes growth until full length. So then we went on to our third and the final part of this work, which is a multi-stressor experiment. And this took kind of findings from the first 2 experiments and is looking to combine them in the multi-stressor setting. So this experiment was set up very similar to the temperature performance curve one. And the treatments were based on the thermal tolerance range that we found in that experiment. So it had the amputation yet again, it had a phenotypic assay, but in addition to the phenotypic assay, we also sampled some key early time points for further histology to look into how the multi-stress aspects are affecting those early stages of regeneration, which sadly hasn't been analyzed yet. So that's another watch this space, basically. In addition to this, we also took more celomocyte counts and did the writing response test again to see what was going on. So the setup was very similar, but using 9 conditions this time. So we had temperature conditions of 8, 14, and 20 degrees Celsius. And we chose these because they were shown to be well within the thermal tolerance range. And we wanted to see how pH alterations would shift this temperature performance curve, basically. The 3 pHs we selected were ambient, which was around 8, then 7.5 and 7.2. And while 7.2 is pretty low, these are a coastal species and they do experience these kind of drastic fluctuations daily. So this is more about the extended period of exposure. Sadly, more conditions means more individual housings and more heterotanks and more everything, making it a little more logistically challenging. pH was dropped using CO2 that we bubbled directly into the heterotanks. We didn't use solenoids, which are very commonplace in acidification work, but they're quite expensive when you get up to 9 conditions. So this makes it much more affordable, and we use gravity-fed flow rates, as well as a constant bubbling of a set amount of CO2. And we found it was actually incredibly stable over the course of the experiment, which was fantastic. So what we found here, I've picked kind of 2 ways of showing this data for now, because I'm still undecided as to what's the way to do it. So the first here is as a percentage length of regenerating appendages compared to homeostatic ones. So we took 10 regenerating from each individual every sampling point, and then 10 measures of homeostatic lengths, and then worked out a percentage of the regrowth. And it clearly shows that growth is pretty consistent over time, though it is slightly varied between individuals. But despite this, we get very clear indication that colder treatments are significantly retarded in their growth rates, and that pH had very limited effects on the rates. The second one here is, yet again, using that growth equation. So this is the temperature performance curves, and it's over the course of that temperature treatments along the x-axis, and those 3 lines represent the 3 pH treatments. And yet again, pH was very limited, but as an impact, but temperature showed a very notable effect. So temperature was shown to be the driving factor here, and the models run on this, so the GLMs also showed no real interaction between pH and temperature as stresses. Despite there being no obvious effects of pH and no real interaction between the stressors in terms of regeneration, there was, and calcification, there was some interesting observations that I've noted and I'm looking into further from now on. So in the reduced treatments, pH of 7.2, the spines were a lot more brittle, very easily broken. They suggested compromised structural integrity, and this was even in the homeostatic spines. And this kind of insinuates that maybe acidification causes the difficulty in calcification, and this makes sense. And although this also suggests that sea urchin would prioritize a full-length spine for protection, even with the sacrifice of kind of structure and strength, which makes sense given how easily they break in these dynamic systems they're in. I mean, why expend energy creating a strong structure that will inevitably be broken anyway? And this has been supported by previous literature. This is from that paper my supervisor was involved with. Incredible SEM images. And you can see like the gaps in the spines are bigger with higher PCO2 levels. So I've also sampled some spines for weight-bearing tests and some scanning electron microscopy. So hopefully I'll find similar stuff. And the other observation I found was a lot of high mortalities in the 20-degree treatments in this third experiment. And this was odd because in the first experiment at 20 and 24 degrees, there was no mortality really observed at all. And I'm assuming seasonality is expected to be the cause here. The temperature performance curve experiment was done in the summer, and this one was in early spring. And these populations are being collected from the wild and acclimated. They're not stock in the aquariums. So I'm expecting that seasonality is altering kind of the thermal tolerance limits for the species. And hints. So in the first temperature performance curve, you can see there, that's roughly where we were. But in the later experiment in spring, I think we've shifted into the population crash territory. So it's expected that seasonality alters these thermal tolerance limits, and that we did actually hit this threshold. Maybe not for the growth rates, but definitely for overall survival. And maybe the celomocytes and stuff will back me up on that. So basically, conclusions so far, the very obvious ones. Unfortunately, I don't quite have the nice little like nuanced conclusions yet because the analysis is still ongoing. But temperature is very clearly a driving factor here in regeneration, and by regeneration, I also mean calcification and tissue regeneration. And there is this risk of surpassing thermal limits, and seasonality also is impacting that. There are no notable impacts of acidification, so reduced pHs, on these regeneration rates, but that is definitely not the whole story. And there should always be care taken measuring like an organism's ability to tolerate acidification purely based on something like calcification rates, because a lot of the previous literature has shown there's a big energetic trade-off to calcifying in these challenging conditions. There was a study I was reading the other day about brittle stars and this muscle wastage. So they were using the muscle as an energy source, but the calcification rates remained the same. So overall, it may have an impact, and I'm hoping to look into that further. Regeneration itself we had not really been visualized in sea urchins before, so it does appear to involve this blastema of stem cells, and I'm hoping to confirm that and fill in the gaps there, but that's also a new finding as far as I can tell. And the last one here is more of the observational thing that I'm hoping to look into, but structure does appear to be compromised in these low pH environments for spines. And I've got these planned tests that are being carried out right now and hopefully analyzed. So I couldn't have done any of this on my own. I'd like to thank these wonderful supervisors I have for the advice, support, and guidance. Helena Renardi and Mike Burrows here at SAMS have been incredible with their help for the experimental setups and the ecology and the understanding and analysis of that work. And Vicky Slay over at the University of Aberdeen for all her kind of ongoing help and knowledge on the regeneration aspects of my work. I'd also quickly just like to shout out to Andrew Matthews and Heather McConnell for the technical help in the aquariums, because I couldn't have done that without them either, for the maintenance and setup in general. So big thanks to them as well. Thanks very much for listening to me. And if you have any questions, don't hesitate.
Amazing. Thank you so much. Always interesting to see talks on ocean acidification. It was something that I was myself really interested in back in my university days, and just how it's almost like the evil twin of climate change. You just don't hear enough about it. We are going to go into the Q&A and discussion now. Again, if you have any questions, you can either write them in the chat, or you can use the raise hand function. You can unmute yourself and ask the question, turn your camera on as well if you would like. Another just quick reminder that this is being recorded and going up onto YouTube. So if you don't want to appear on YouTube, please feel free to put your question in the chat. I'm going to start off. So obviously your talk title caught me a little bit off guard when I first read it and when you first sent it to me, wondering where this was going to go, which leads me to my very first question. How on earth do you shave a sea urchin?
Very un-high-techly. A pair of dissection scissors, basically underwater, so they're not pinging off everywhere. They're quite small. They're like that big. And yeah, it's basically a pair of dissection scissors. You've got these rows of tube feet, so they're clearly kind of defined sections. You chop off the spines, you chop off the tube feet, you put it under the water, you let it chill out a bit and relax, so it'll stick out any tube, because the tube feet get pulled in and out. That's the bit that makes it tricky. You let them relax, and then you come back and get the ones you missed. And then you kind of just keep doing that all day until you're pretty confident you've got most of them. But the only trick there is to not damage the epithelial layer, because they've got this test that's not actually an exoskeleton. It's got this thin layer epithelial cells on top. And if you damage that, then it slows down the whole process. So it makes like this analysis of regeneration rate a lot trickier. So I've done that a few times. But yeah.
So how then, just sorry, still on this topic of shaving sea urchins. So how delicate is this as a, how delicate is this? So how, because I'm guessing if you go too deep or too far, you potentially risk damaging that individual, which may lead to complications with what you're testing for. So how much of this is something through trial and error, or how much of this is actually, you know, exactly what you're doing and where to actually snip?
So this was published by my supervisor before I started as a method she'd used before in that paper that was in that presentation. So they developed this themselves. So that it wasn't, thankfully I wasn't coming in starting from nothing, but I did originally I damaged the epithelial layer, and I mean that regenerates too, but it does do it a lot slower. And because we're doing averages of appendage length, it's the first week is always a bit messy anyway, because it depends very much on how you cut them. But basically it was found that keeping it a little longer is better. That gets balanced out in these replicates, and in the repetition, even on that one individual, when you're doing the assay, when I'm like writing down the length of this one and this one and this one, I'm like, this data looks so messy. Like it looks like it's all, and then when you actually come to analyze it, it's pretty surprisingly robust. It can survive, doing things slightly differently. We took it to be, I should do, like, if I'm amputating this section, I should do every individual in this experiment so that it's all uniform to how I've been doing it, because everyone seems to do it a bit different. Because it's not high tech, it's just me going in with a pair of scissors. So in that way, the replication and me doing all of that on my own, or someone else doing all of that on their own, keeps it surprisingly robust. It works very nicely, but it's not high tech.
Just had this image when you brought it up of just a whole barbershop type Delio. So moving on to some probably more questions based on the talk itself. So obviously you're working with this particular sea urchin, and I myself, I mean, I said this to you before, I know sea urchins are, of course, a living organism, but it's so weird to view them that way just by how they look and how they sort of move around. Obviously, there's a lot of different species of sea urchins, and they exist from the depths of the ocean to the one that you're working on, which I believe is more intertidal. Is that correct, or is it also a deep sea?
Very coastal.
So how do, and obviously you've not looked at other species, but is this something that you think will affect sea urchins as a whole, or will habitat and depth and things like this impact sea urchins differently in that regeneration?
So I only work on one species, but here at SAMS, and certainly previous work with my supervisors done with other students and stuff, is looking at a couple of these other species. We have 3 stock species here. Echinus, the big kind of palo ones that live really deep, the Psammechinus are my ones, and the Paracentrotus are the kind of middly purple ones that maybe everyone's a little more familiar with. And they're kind of in the middle of the deep sea and the coastal. So we do have this nice range that is being looked at. I'm doing this coastal one because they are experiencing this, like with the tides, dramatic, temperature changes every day, twice a day, and things like that. And certainly in terms of temperature, the deeper they go, maybe the more stable temperatures, the sea surface temperatures is what we often talk about. So the deeper seas are a little more tolerant of changes. It's still, I'm sure, shifting, but to lesser extents than sort of this is going to be the first kind of interaction of these increasing temperatures and pH. But in terms of, this, if this stressor is exposed to any of them, I'm sure they do seem to react in very similar ways. The calcification process is the same. Regeneration, it's still quite conserved across these species. So this is just a convenient model for me, but it should be representative of quite a lot. For me, I can just go pick them up at low tide, so it's great.
Yeah, that's always a benefit when you're working with animals. It's easy to get your hands on them, though I don't know if you would actually want to put your hands on them. Though I did see the photo of your supervisor holding one, which, as someone who used to dive, I was always told to be more scared of sea urchins than anything else. So obviously, temperatures, pH, this is changing now. I think I can't remember if we've hit or surpassed the 1.5 degrees global temperature change. I'm not sure what that is with ocean acidification, how big that change has happened over the last few decades. So this is clearly something that's happening right now. So with your work that you're currently doing, is this a case of, well, this is happening now, we need to see what the effect is, or is it more of a case of this will be happening soon and we need to see potential what ways that we can slow that down or prevent it or increase adaptation towards it?
So I'm not necessarily looking at like how to prevent it. Like I'm not, combating climate change isn't a thing I'm working on. I'm very much on the understanding it side of things. I mean, but the conditions we're choosing for these experiments kind of go both ways. Like, so we've got these conditions we're in now, and then near future conditions, and then like by the low extremes, like 2000, 2100 sort of ballpark figure from some of these worst case prediction scenarios from some of these experts. But certainly the work I'm doing right now is more like understanding. Like the more we know about how, in this case, sea urchins, but any organism is surviving, adapting, or not, the better we can understand what the risks are and how best to kind of go about protecting that. What the kind of priorities are. Like it's tricky to protect against everything all at once. So if we know what the main factors are, we can know what we're looking at and how to help with that. Obviously, sea urchins are a really small part of like an ecosystem, but they are a key species. They're very heavy algae grazers and things like that. They're very much part of the trophic chain, and so even if sometimes they're kind of overlooked in terms of how vital they are important to an ecosystem. Um, so yeah, it's just about, for me, understanding the coping mechanisms, how they adapt, and who knows what comes out of the work. I was mentioning to you earlier about this nerve regeneration. Very much not a part of why I started this work, but they can regenerate nerves, and we're looking into kind of antibody staining and stuff to see how nerves regenerate. And if we understand that, maybe someone a lot more specialized than me in something like biomedical or something, who knows, can take what I'm finding from a completely different way of viewing a problem and do something incredible with that too. It's really tricky to predict kind of where things will go when you're doing it almost for the understanding sake. But yeah, I'm not an ecologist, so I may have brushed over a lot of.
I mean, it's an important. I mean, a question I would sometimes get asked, because I did dip my toes in research, and my area that I dipped into was ocean acidification on the effect of chemical communication in oceans. And I was often asked the question, like, realistically, are animals going to be able to adapt to these changes? Are we going to see evolution change within these animals? Which is always a hard question to ask, because a lot of what we're kind of doing and a lot of where research does tend to be is actually trying to understand it. And yeah, that's kind of moves into the next question. So obviously you're very experienced with the work that you did. You said for the UN in Monaco, which must have been an incredible experience. So if we, and this could potentially be a really horrible question, so I do apologize ahead of time. The future of your work, the where it's going, where do you see sort of the wider implications and impact of your research? And ideally, if you could choose, where would you continue it on to next?
Yeah, it's not the easiest question.
Yeah.
But yeah, I mean, it's kind of what I was saying before. Like for me, very much it's this understanding, and then hopefully understanding for other people too. I'm not involved in policy making or anything like that, but I do feel like we can tell people this is happening and it's bad, but we need concrete proof before people start listening. So I mean, the more proof we have, the better, sort of thing. And yeah, as you were saying about coping and adapting, that's really tricky too. As I mentioned, like we don't really understand like multi-generational exposure risks, and my sea urchins came from the environment just outside of here and came straight into these conditions. But if they've been raised in these conditions, are they more tolerant? Do they do any of this adaptation themselves? So that's already tricky to know. And in terms of where I want it to go and where I want to take it, I'd like to, obviously PhD, very finite time frame to do this stuff that you've come to do and get done hopefully well. But kind of in the future, I'd like to take what I've done in different ways and bring it all together with different species, get a real understanding of how, like maybe in a single environment, in a single kind of trophic chain, how this is being impacted by, say, OA and stuff, because sea urchins are not living at 24 degrees, and maybe that's because the algae's not there or the nutrients aren't there, or maybe it's because they're stressed too much. And it's so complicated when you start bringing the fact that all these species live together into the mix. I'm looking at a single species and it's already complicated, and then when you bring in the fact that all these other species are having exactly the same impact and all these challenges, it's isn't possible. Like I don't know where it will go. It's probably going to be a case of I find a rabbit hole of something I find interesting, and then I end up going down that instead of where I thought I was going to go, because it's such a big thing, and it's only really multi-stressors in particular have only kind of recently come into the limelight. It's now that we've accepted that, you know, this is happening and it's bad. So I know there's loads of people working on it now, and I think standardizing and bringing it all together is a big thing that would be nice. We're all doing our work, but we're all doing it in different ways. We're picking different conditions. We're in different species. We're in different climates. How do you unify all that understanding? It's not something I can answer easily, put it that way.
So I've just been made aware that we are getting questions in the Q&A. Unfortunately, I didn't have it open, so I haven't seen. So I'm going to cover some of the questions that we've got coming in. I'm not going to read people's names out because, again, this is being recorded. I don't want to be doing that. But the first question, and we're getting a lot of compliments about the talk as well, which is always nice to see. So the first question here is, I was wondering if you could go over into detail about food supply to the urchins, where were they well fed throughout the experiment, and do you think that might offset the effects of ocean acidification?
It's definitely a factor. So for me, I didn't want it to be a factor specifically because of that. I've already got these multi-stressors of temperature and pH. So food availability is a stressor in itself. That's a third stressor. That's a what, 27 treatments if you actually include it properly. So I made sure they were always fed. They always had fresh sort of algae. We just chopped off a square, put it in a little tank. They always had that available to them so that it was not stressing them. Because even if I don't know if it would affect OA. That's a study in itself. Maybe I'll do that at some point. I don't know. But certainly I didn't want it to be a stress because, you know, there's still these immune responses. And I mentioned briefly about energetic costs of maintaining the structures. So if you reduce the food, they should have less energy. Maybe the calcification is impacted when they're calcifying in these challenging conditions, where it costs a lot more energy even just to maintain a skeleton or a spine in this case. So yeah, they were ad-lib fed consistently throughout the whole thing because I didn't want that to confound anything, basically.
That is an interesting point, though. Like, because I'm guessing if we're talking about this in environmental models, oceanification, climate change is going to be affecting everything. So if food sources are limited, that's very interesting.
Mm. That's why it gets so complicated. Yeah, it's crazy.
Another question. So, do you think that your findings can be extrapolated to inform conservation efforts aimed at mitigating the impacts of climate change / acidification on other marine species? I think we covered this a little bit, but I don't know if there's anything you'd like to add there.
I mean, it's what I was saying before. Like all these species react. There is conservation. Like there's quite conserved reactions across a lot of species, depending on how they're calcifying or how they're interacting with the environment. So I mean, whatever is found in sea urchins is not only going to be found in sea urchins. It'll be found in other species. They do use quite a unique calcite source. This magnesium calcite is not always used. Often it's like aragonite in corals or calcite, regular calcite in a lot of other species. So there will be some impact. But calcification as a process is pretty tightly conserved across species usually. Different cells do it, but it's the same basic function. And same with regeneration, like that blastema I was mentioning is very common in a lot of regeneration work. Whether it can, for informing conservation efforts, I don't know if my stuff on its own would do anything. I think it's when this research kind of comes together and you get this real big picture you start developing this big picture of how things are working and interacting with each other and what is most at risk and what to prioritize looking after and things like that. Obviously, coral reefs were very big. This is happening now, preserve them. And there's been some really good research and really good work into restoring them and helping that. So I think my stuff on its own maybe won't make much headway in the world. But when you start, if everyone contributes a little bit and you start getting these nice lit reviews and things like that are really compiling all this data, I think that's when you can maybe start using it to kind of inform policies and things like that.
Yeah, it's because I don't know, because I know obviously there's a lot of species that we could say give us a good indication of environmental health. But again, in this, as you say, trying to just say this one species will tell us about, especially when every species does things differently. The organisms I worked on were crabs, which obviously calcify slightly differently. And I can't remember actually. So the work, as I said, I did was on chemical communication, but I do remember seeing things on calcification for that. Yeah, it is. It would be interesting to sort of see that kind of a metadata study looking at all these. Oh, another question. So, do you know roughly what concentration of CO2 parts per million you would use to achieve each pH treatment? Which is the most realistic as a treatment for the end of the century?
So that is a watch this space thing. I didn't in spot checking, it's much easier to spot check using pH. So I've got all these water samples that I'm now analyzing. None of that is quick. So I've got a load of water samples, like a time series throughout each of these experiments. Well, certainly the multi-stressor CO2 experiment. I have to analyze for dissolved inorganic carbon and total alkalinity, and then I'll have this, because obviously when the research comes out, that's how I should be phrasing it, as parts per million of PCO2. I think the most realistic is going to be that mid one, that 7.5. That's kind of why we picked it for the end of the century. The 7.2 is very extreme, but that was chosen on purpose as a very extreme stressor, and because of this coastal kind of dynamic where they're coming out of the water a lot and getting some weird exposures. That's a watch this space.
As is, I think, a lot for these sort of research areas. We have time for one last question, and again, it kind of touches upon something we've already discussed, which is wondering whether your data could be used to draw a big picture conclusions for ecosystem-wide impacts down the line. And I guess this is kind of the big question: can yours and other people's works be brought together? I don't want to say the word how likely is that, but is this something? Obviously, we have numerous different conferences that happen throughout the year. Is this something of a wider conversation that you know of, or would encourage, or big picture?
My stuff on its own, if reading the question initially, I'm like, no. But as you say, once everyone starts chipping in, then yes. I mean, OA itself, acidification itself, without the multi-stressors even, has become like a lot more of a relevant topic. Like years ago, it wasn't really a thing. It was a thing for a bit. Microplastics came on the scene. That took a lot of the attention because it was the new thing. And I mean, obviously, I'm not saying microplastics aren't important, but it did shift focus away from this as well. And I think it's kind of now found its middle ground between all these kind of very obvious environmental issues. Like, I don't think anything's stealing the limelight as such. So there's a lot of OA work now. I know there's like this these networks now for ocean acidification. There was one based in Monaco when I was there, and they're looking into this collaboration worldwide, this global collaboration, and helping like developing countries in assessing their own conditions and things like that. And it's a really good start for like standardizing how we're approaching this work. And I think if we want to bring it together, we have to standardize how we approach this work. It's impossible to kind of draw conclusions when everyone's doing their own thing. But I'd say I'm hopeful that. I wouldn't say it's all doom and gloom. I think I'm hopeful that everyone in this line of work, like doing this kind of research, is doing it for the same reason. We're doing it for the same goal. We want to understand what's going on and hopefully help inform how to potentially stop it. So, you know, collaboration's always a thing, and I think everyone's quite keen on doing that. So I'm hoping my data can be used in that. I'd love to be involved in it. I don't think it's wasted data. So yeah, I'm hopeful. Let's go with hopeful.
Awesome. So we are coming up to time now. So I'm just going to end the webinar firstly by saying thank you once again, Angus Taylor, for your amazing talk. It's been really, really interesting. And I'm actually like, yeah, I've really enjoyed myself, and I hope everyone who's watched it today goes away with more information about shaving sea urchins at the very least. I'm sure there is a lot more takeaways than just that. I'd also like to mention that there has been a link posted to an article, a paper that was published last year on into ocean acidification. So that's been posted in the comments if people would like to read more around this topic. I'm also going to post the link to the third Delve Deeper webinar series, which is coming into the comments now. So next time we are going to be diving into the intricate world of molluscan biology with Dr. Carmen McDougall, whose talk will span from pearl formation to shellfish reef restoration, showcasing vital roles into cutting-edge molecular techniques in advancing shellfish aquaculture and conservation efforts. As again, that link is in the comments, and hopefully we'll see you next time. So thank you everyone once again for joining us, and that brings us to the end. So I'm going to stop the recording now. Thank you very much.